Good afternoon, dear colleagues. Today we will start our webinar concerning on drug resistance mechanisms in therapeutic context. This topic is very important because we now can notice uh, huge burden for healthcare system and problems with antibiotic resistance, with problems tuberculosis resistance drugs, because problems related on with resistance of bacteria and problems it's now huge. For example, malaria is resistant for every antibiotic in the world, and IC and pneumococ can be get resistant to for antibiotics. But not just antibiotics and antiviral and antifungal drugs can have resistance, but and chemical drugs can have resistance. It can be, we will talk about today drug resistant in neurology, drug resistant psychiatry, drug cardiology, huge problems, clopidogrel resistance, aspirin resistance in patients, and it's very actual problem right now in clinical practice. For our new participants, I would like to introduce myself. My name is Galina Cordero. I am QPPV head of pharmacovigilance department, clinical expert, DCK Pharmac. I conducted webinars in LinkedIn and YouTube channels, and I will be very thankful if you will subscribe to my YouTube channel, Galina Drug Safety from Galina Cordero, and visit my website, which I promote and add new information in my site too. This site will get some transformation. It will be modern site with modern actual information concerning all medicine problems, perspectives, challenges in treatment. I think it will be useful for you in your daily practice. Problems drug resistance is not new, and it's defined as a reduction of loss of drug efficacy despite appropriate dosing spans multiply therapeutic areas including infection disease, oncology, cardiology, and psychiatry. The global burden and resistance related treatment failure underscores the need to delineate the underlying mechanism, identify responsible agents, and develop sustainable management approaches, according Ventala 2020. Main mechanism of drug resistance is genetic and epigenetic changes, including alteration in genes and epigenetic regulation. Example can be doxorubicin, methotrexate. It can be efflux transporters and drug metabolism, overexpression of efflux transporters of metabolic enzymes. An example is aspirin. Altered drug targets, modification or mutation of drug targets, example aspirin, and adaptation and survival, cellular adaptation and resistance to cell death, example can be cisplatin. If we were talking about general mechanism of antimicrobial and pharmacology resistance, we can notice K mechanism. First of all, is target modification, mutation of PBPC, ribosomal subunits, and kinases. Drugs affected can be penicillins, macrolides, TKIs, for example, this can be EGFR, ABCR, ABL. Efflux pump overexpression It can be examples P-glycoprotein, BCRP, MRP transporters. It can be drugs affected doxorubicin, ciprofloxacin, and carbamazepine. Enzymatic degradation, example of this mechanism is beta-lactamase, acetyltransferases, and drugs affected cephalosporins and aminoglycosides. Biofilm formation can be protective microbial community structures, fluconazole antibiotics in Proteus aeruginosa. Alter drug metabolism, cytochrome 450 induction or suppression. Drugs affected can be midazolam, warfarin, and antiretroviral. And reduced uptakes, it can be purine channel loss, OPRD, and Proteus aeruginosa. It can be PNAM and fluoroquinones. When we talk about more detailed the mechanism of drug resistance, and we will start from genetic and epigenetic modification. It can be mutation target genes, EGFR, BCR, ABL, and well characterized in oncology and contribute to primary and acquired resistance. Epigenetic regulations such as DNA methylation and histone modification also play a central role in down-regulated drug targets or upregulated efflux pumps, according to Zhou et al. 2023. And these factors you see can lead to drug resistance at all. When we're talking about genetic alterations such as point mutation, gene amplification, chromosomal rearrangement, more detailed in K oncogenes and drug targets, again, it can be EFG, VR, BCR, IBL, ILK, and well-established contributors to both primary and acquired resistance, particularly in cancer therapy. For example, the T79OM mutation in EGFR confers resistance to first-generation tyrosine kinase inhibitors in non-small cell lung cancer, while BCRP-ABL mutation underlie resistance in chronic myeloid leukemia despite imatinib therapy, according Hussman et al. 2020. This mutation alters the structure of drug-binding domains, reducing affinity and efficacy. In parallel, epigenetic mechanisms such as DNA methylation, histone acylation, deacetylation, and not coding RNA mediated regulation also significantly influence drug response. Hypermethylation of promoter regions can silence tumor suppressor genes or downregulate pro-apoptotic factors, while histone modification may lead to chromatin remodeling that affects transcription of drug-resistant genes. Furthermore, epigenetic upregulation of ABCB1 encoding P-glycoprotein has been linked to enhanced efflux of chemotherapeutics, contributing to multi-drug resistance, according to Zhou 2023. Efflux transporters and drug metabolism. Overexpression of ATP binding cassette ABS transporters, including P-glycoprotein, PGP, enhances drug efflux and lowers intracellular concentration of chemotherapeutics and antiepileptics, according to Sarkady et al. 2022. Upregulation of cytochrome 450 enzymes modifies drug metabolism and may induce resistance through altered pharmacokinetic, according to Rostami Hajijan 2021. A key mechanism of pharmacology resistance, when we talk about flux transport and drug metabolism and drug resistance more detailed, involves the overexpression of ATP-binded cassette ABC transporters, a superfamily of membrane proteins that actively expel a wide range of xenobiotic from cells using ATP hydrolysis. Among these, P-glycoprotein PGP-ABCB1, multidrug resistant associated proteins MRPC ABCCS, and breast cancer resistant protein BCRP ABCG2 play dominant roles in reduced intracellular drug accumulation. Overexpression of P drug, for instance, has been shown to confer resistance to structurally unrelated anticancer agents such as doxorubicin, paclitaxel, and vincristine, as well as several classes of antiepileptic and antibiotics, Sarkadi et al. 2022. These transporters are often upregulated in cancer stem cells as the blood-brain barrier, limiting the efficacy of chemotherapeutics and CNS active drugs. In parallel, altered drug metabolism, particularly via cytochrome 400 P450 enzyme system, can significantly affect drug pharmacokinetics. Induction of enzymes such as cytochrome P3A4, cytochrome 2C9, and cytochrome 2D6 may enhance biotransformation of active drugs into inactive metabolites, thereby reducing their therapeutic concentration. Conversely, inactivation of prodrug may be impaired. This metabolic adaptation can occur in tumor microenvironments, liver tissues, or intestinal epithelia, often mediated by transcription regulators such as pregnan X receptor PXR and constitutive androsten receptor CAR, which are known to co-regulate both drug metabolized enzymes and efflux transporter genes, according to Rustami Khodijan 2021. The coordinated upregulation of efflux transporters and metabolic enzymes contributes to a multidimensional barrier against drug efficacy, leading to multidrug resistance and treatment failure. Understanding and targeting this mechanism via efflux pump inhibitors or enzyme modulators remains a major focus of translation research in oncology, infection disease, and neurology. Additional examples include kinase inhibitors, imatinib, showing reducing efficacy due to mutation in the ATP-binded pocket of BCR-ABL or AGFR, T3151 and T79 or M mutation, respectively. Antibiotic resistance, where mutation in ribosomal proteins or DNA derase reduce binding affinity of macrolides or fluoroquinolones. This alteration not only impair drug target interaction but may also shift allosteric regulation or enzyme kinetics, contributing to both primary resistance, inherent insensitivity, and acquired resistance after initial therapeutic success. Such adaptive changes emphasize the need for next generation inhibitors that can bind alternative domains, allosteric sites, or mutated targets, as well as for companion diagnostic to personalize therapy based on target mutation profiles. Altered drug targets structural alteration in binding sites, for example, COX1 in aspirin resistance or beta-bulin in taxane resistance, impaired drug target interaction, Patron et al. 2021. Other drug targets and binding affinity and resistor mechanism. A fundamental mechanism of drug resistance involves structural modification pharmacological targets which reduce drug binding affinity and compromise therapeutic efficacy. These changes can be the result of point mutation, alternative splicing, post-translation modification, or conformation shift in the molecular structure of target proteins. For example, in aspirin resistance, alteration in the cyclooxygenase SOC1 enzyme, including polymorphism or conformation changes near the active serine residue, SER 529, can impair irreversible acylation by acetylic acid, reducing inhibition of thromboxane A2 synthesis and diminishing antiplatelet activity concerning on according Patrona et al. 2021. This has major clinical implications for secondary cardiovascular prevention. Similarly, resistance to taxane-based chemotherapeutics, paclitaxel, docetaxel in cancer such as breast and ovarian cancer is frequently associated with mutation in the beta-tubulin gene, tubulin B1. This mutation alters the drug binding domain of microtubules, preventing taxane stabilization of microtubule assembly, thereby allowing continued mitotic progression and cell proliferation despite drug exposure, according Johnson 2023. Additional examples include kinase inhibitors, imatinib, antibiotic resistance, and such adaptive changing emphasize the need for next generation inhibitors that can bind alternative domains, allosterotic sites, or mutated targets, as for companion diagnostic personalized therapy based on target mutation profiles. If you see on these pictures um, algorithm how it's really working from for example normal binding site and mutation when we're talking about cyclooxygenase gene 1 according mutant SERF 529. You see how it's working and when you understand this mechanism, you can extrapolate this knowledge on drug resistance because, as we see, not just for antibiotics and for antiviral or fungal, antifungal, or for example, anti-tuberculosis drugs. In these classes associated with viruses. It can be presented antibiotic drug resistance and in other drugs. For example, drugs commonly associated with resistance include tyrosine kinase inhibitors. Examples can be imatinib, erlotinib, gefitinib, and mechanism of resistance, a target mutation, BCR-ABL, T3151, EGFR, T79OM activation or bypass pathways. Platinum-based drugs, cisplatin, carboplatin, increase the DNA repair, its mechanism of resistance, efflux via MERP2 detoxification by glutathione. Taxanes, paclitaxel, docetaxel, mechanism of resistance, it can be beta-tubulin mutation, P-glycoprotein overexpression. Anthracyclines, doxorubicin, daunorubicin. It can be topoisomerase 2 alteration, increases drug efflux. Hormonal agents, tamoxifen, abiraterone, receptor mutation coactivator dysregulation, and immune checkpoint inhibitors, nivolumab, pembrolizumab, PDL1 upregulation can be as a mechanism of resistance. Antimicrobial, it's include approximately all classes of antibiotics. If we see, for example, how across which mechanism realized antibiotic resistance. Beta-lactams, examples penicillin, ceftriaxone, beta-lactamase production altered BPS. Fluoroquinolones, ciprofloxacin, levofloxacin, mechanism of resistance, DNA, gyrase, topoisomerase and mutation, efflux pumps, macrolides, erythromycin, azithromycin, mechanism of resistance, ribosomal methylation, efflux. Amylglycosides, gentamicin, amikacin, included mechanism enzymatic inactivation, target modification. Tetracycline, doxycycline include ribosomal protection proteins efflux. Vancomycin, mechanism of resistance, DLA target modification in VRA, and rifampicin, rifampin RNA polymerase mutation across B gene. Antiviral drugs for these classes characterize resistant to for antivirals. It include HIV antiretrovirus, zidovudine, efavirenz, dolutegravir. Mechanism of resistance is RT protease of integrate mutation. RCV antivirus, sofosbuvir, ledipasvir, NSPA, NSPB mutation. Influenza antiviral, oseltamivir, Tamiflu, neuraminidase mutation, and herpes viral drugs, acyclovir and anciclovir, thymidine kinase or DNA polymerase mutation. Why resistance is important for pharmacovigilance and for clinical practice? Because absence of efficacy can be characterized for patients and therapeutic inefficacy. It's why you should remember that drug resistant and can be difference where drug tolerance can be cause of therapeutic inefficacy in patients. It's why this knowledge are very important. Antifungals, azoles as a drug class, exams fluconazole, itraconazole, mechanism of resistance, its ERG111 mutation, efflux pump activation. Echinocandins, caspofungin, FKC gene mutation, glucan synthase. Polynase, amphotericin B, reduced ergosterol content. Neurologic psychiatric drugs. It include antiepileptic drugs, phenytoin, carbazepine. Mechanism of resistance can be through PGP overexpression, sodium channel mutation. Antidepressants, CCRIs, and tricyclic antidepressant alter serotonin transporter receptor desensitization. Antipsychotic drugs, haloperidol, clozapine, dopamine receptor upregulation or modification. Antibiotic-resistant pathogen VCO priority list. It include Mycobacteria tuberculosis, rifampin, isoniazid drugs, Pseudomonas aeruginosa, carbapenems, and aminoglycosides, Acinetobacter baumannii, colistin, carbapenems, Enterococcus fecalis, vancomycin, Staphylococcus aureus, methicillin, clindamycin, Klebsiella pneumonia, carbapenems, SABC drugs. Drugs resistant in the cardiology is characterized to a very important while because cardiology diseases can be very dangerous for patients and absence of efficacy because of drug resistance can be cause of drug. For example, antiplatelet agents very important classes aspirin and clopidogrel. Aspirin through SOX1 gene polymorphism increased platelet turnover and coating reducing absorption can be and clopidogrel cross mechanism tachrom TIP2 C19 loss of function alleles tachrom 2 C19.2.3 and P2Y12 receptor polymorphism according Media and 2020. You can attest influence of diseases too. For example, there are information from open source that clopidogrel sometimes is not effective in patients with diabetes mellitus. Statins, atorvastatin, simvastatin, mechanism of resistance, LCO 181 gene polymorphism, affecting hepatic uptake, increasing HMG GOA reductase activity, poor adherence, according Link et al. 2022. Beta blockers, metoprolol, bisoprolol. This drug can have drug resistance too through ADRB1 polymorphism, ARG389 GLI, altered beta adrenergic receptor sensitivity, according Batangar et al. 2021. AC1 inhibitor AC or RBC like enalapril and losartan. Polymorphism in ICA of angiotensin receptor genes, RIS pathway hyperactivity, renal handling variation. Diuretics, furosemide hydrochlorothiazide, distal tubular adaptation, RIS activation, epithelial natrium plus channel mutation. Clinical implication of cardiovascular drug resistance. It can include aspirin resistance, increased risk of recurrent thrombotic events, including myocardial infarction and stroke, clopidogrel resistance associated with stent thrombosis and failure of dual antiplatelet therapy DIPT in acute coronary syndrome. Statin hyperresponsiveness, poor LDL-C lowering, suboptimal cardiovascular risk reduction, beta blocker ineffectiveness, inadequate heart rate control, persistent angina or arrhythmias, diuretic resistance, persistent edema, and uncontrolled hypertension and heart failure. Monitoring and diagnostic tools include for diagnostic drug resistant cardiology include platelet function test, verify now PFA 100, thromboelastography to assess antiplatelet resistance, genotyping, cytochrome 2C19, SLC01B1, and ADRB1, polymorphism to git therapy, clopidogrel or ticagrelor. Lipid panels and ApoB evaluate statin efficacy, and BNP and T-proBNP in fluid balance assess response to diuretics. Pharmacovigilance and regulatory consideration. Pharmacovigilance and regulatory consideration include FDA includes cytochrome 2C19 biomarker for clopidogrel. Signal detection and adverse reaction associated with ineffective therapy. For example, it can be MI despite antiplatelets should be evaluated for resistance. Risk minimization measures, recommendation for genotyping in higher risk population. Periodic safety update reports and PREBARES updates include emerging and resistant data, therapeutic failures, and real-world effectiveness. Drug resistance in neurology. It includes disorders, epilepsy, depression, Parkinson's disease, multiple sclerosis, and migraine, and its covering drug class involved antiepileptic drugs, ADC, phenytoin, carbamazepine, and levetiracetam. Mechanism of expression of P-glycoprotein at the blood-brain barrier. Mutation in sodium, calcium, and GABA receptor genes alter drug metabolism, cytochrome 2C9, cytochrome 2C19 variants. Depression, CCRIC, CNRC, ETCAs, mechanism of resistance of this group drug classes cover downregulation or desensitization of serotonin receptors 5-HT1A and 5-HT2A. Genetic polymorphism in serotonin transportase CLC6A4 and inflammatory cytokine-induced resistant interleukin 6 and tumor necrotic factor alpha. Parkinson's disease, levodopa dopamine agonist. Mechanism of resistance covering dopaminergic receptor downregulation, loss of presynaptic dopamine neurons, and peripheral metabolism via COMT and MAO-B pathways. Multiple sclerosis, drug class involved immunomodulators, EFNB, and glatiramer acetate. Mechanism of resistance, drug resistance include development of neutralized antibodies, immune escape mechanism, migraine, triptans, CGPR inhibitors, receptor desensitization, 5-HT1 flash anti-B and 1D upregulation of CGPR pathway and genetic variability in response pathway. Clinical impact on patient safety and outcomes. Drug-resistant epilepsy DRE occurs up to 30-40% of patients, increases the risk of sudden unexpected death in epilepsy, injuries, and psychosocial burden. Treatment-resistant depression is associated with higher suicide risk, chronic disability, and reducing life expectancy. Levodopa resistance or complication despite therapy, signal resistant, require organ review. Diagnostic and monitoring approaches. Pharmacogenomic testing, cytochrome 2C19, SCN1A, COMT to get therapy in epilepsy, depression, and Parkinson. PGP expression assays as the BBP, BBB, experimental limited to research setting. Therapeutic drug monitoring (TDM), essential for IEDC like valproate, carbamazepine, neuroimaging and EEG monitoring for assessing structural and functional causes on pharmacoresistance, pharmacovigilance and regulatory implication in drug resistance in neurology include signal detection, unexpected worsening, non-response of adverse outcomes under standard therapy should be evaluated as potential resistance signals. Risk minimization strategies, inclusion of genotyped recommendation SMPC, included cytochrome 2C19 for escitalopram, switching to non-PGP substrates lacosamide and epilepsy. Regulatory guidance, including European Medicines Agency, Food Drug Administration, required resistance-related data and periodic safety update reports, risk manager plan for central nervous system drugs, inclusion of resistance risk and benefit risk evaluation, emerging regulatory trends, integration of artificial intelligence-based predictive tool for treatment response and real-world effectiveness monitoring in neurological drug development, drug resistance in psychiatry, major depressive disorder, including classes CCRIC, CNRIC, TCA in mouse, include SK mechanism of resistance, polymorphism in SLC6A4 serotonin transporter gene, 5-HT receptor desensitization downregulation, 5-HT1A, overactivation, OSPA excess and inflammation, and you should remember that in patient with post-traumatic disorder, in patient with major depressive disorder, parallel with activation, overactivation of PA axis, very intensive going processes, inflammation, and including chronic inflammation, even parodontitis, periodontitis, and processes, all chronic inflammation which via as a sleeping processes, parallel activated metabolic disorders, and very high risk of carcinogenesis because in this group of patients with major depressive disorders and in patients with post-traumatic disorder syndrome, very weak immune response. It's mean including response like anti-tumor immune response. It's why you should pay attention in this group major depressive disorders patient. Despite of suicide income, it's very as I said, group which you should pay attention on this patient. Plus, in this group, impaired neuroplasticity and reduce it as a marker. You can include too as a marker is BDNF expression. Bipolar disorders, mood stabilizers, lithium valproate, key mechanism of resistance in this group is GCK3B pathway dysregulation, mitochondrial dysfunction and oxidative stress, and genetic variants and INK3, CICNA1C. Schizophrenia, antipsychotic, typical, atypical, Key mechanism of drug resistance in these groups is dopamine D2 receptor supersensitivity for internalization, polymorphism in DIR2 and SMT, glutamatergic NMDA receptor dysfunction, overexpression of P-glycoprotein as the BBB, anxiety disorders, including drug classes SSRIC and benzodiazepines, GABA receptor desensitization, JABRA1 polymorphism, hyperactive amygdala circuits and SPA axis dysregulation, and serotonergic dysregulation, HTR2A classes mechanism. It's very important and you should remember it. Clinical impact on patient safety and treatment outcomes. Treatment resistant depression affects 20-30% of patients as linked to higher suicide rates, impaired social functioning, and chronic relapse. Clozapine resistant schizophrenia present major challenges: poor functional risk, violence, hospitalization, and comorbid substance use. Failure benzodiazepines or SSRIC and anxiety increases somatic symptom burden and the likelihood of polypharmacy. Increases side effect burden. Higher doses of polytherapy and resistant cases increase the risk of weight gain, metabolic syndrome, QT prolongation, neurotoxicity, and sudden death. There are results of researches which annotated this tragic outcome as a sudden death, more characterized for tricyclic antidepressant. Diagnostic and therapeutic tools for resistance, pharmacogenetic testing, FDA and EMA endorse cytochromic 2D6 and cytochromic 2C19 genotyping to personalize CCRI and antipsychotic therapy. Neuroimaging biomarkers, fMRI and PET, evaluate functional resistant patterns like amygdala hyperactivity in anxiety. AG and QEG, EEG used in psychiatry research to detect neurophysiological correlates of resistance. Biomarkers as BDNF, inflammatory cytokines, including interleukin 6 autoreactive protein, correlate with antidepressant resistance too. Pharmacovigilance and regulatory implication. Signal detection, non-response, or paradoxical reaction may represent resistance rather than adverse reaction, especially in black box situations, suicide risk under CCRIC. Risk minimization, pharmacogenomic labeling, cytochrome 2C19 for escitalopram, cytochrome 2D6 for aripiprazole. Stepwise algorithm for switching, augmentation, and non-pharmacologic intervention, ECT and RTMC. RMP, risk management plan, and periodic safety of data report. Reporting therapeutic failure trends in Tier D and Tier C treatment resistant schizophrenia. Real world evidence collection on long term psychiatric medication effectiveness. European Medicine Agency and Food Drug Guidance support resistance research as a part of psychiatric drug development and life cycle safety evaluation. Understanding how drugs lead to resistance in pathogens. Mechanism, enzymatic inactivation, description can be that bacteria produce enzymes that degrade or modify antibiotics. For example, can be beta-lactamase, SBLC, KPC, and aminoglycoside modifying enzymes. Target site modification, mutation, or methylation of drug targets reduce binding affinity. Altered PBPC, penicillin, DNase, GARAS mutation, fluoroquinolones, 23 as rRNA methylation macrolides, efflux pump overexpression, increased active transport of drugs out of the cell, Acar A-B toll system, gram-negative bacilli, methyl macrolide resistant, reduced permeability, loss or alteration of parins reduced drug entry, OPRD downregulation, and Pseudomonas aeruginosa, carbapenem resistance, bypass pathways, alternative metabolic pathway compensate for the inhibited one. VNA-mediated D-ALA D-LAC synthesis in vancomycin resistant enterococci. Biofilm formation, microbial communities resist antibiotic penetration and host immunity. Chronic P. aeruginosa infection in cystic fibrosis, Staphylococcus epidermidis or on catheters. Horizontal gene transfer, SGT, spread of resistant genes via plasmins, transposons, integrons. NDM1 carbapenase gene, MACRE1 colistin resistant gene. Mechanism of bacterial resistance of antibiotic, if we will assume, should include understanding genetic mutation in bacteria. Genetic mutation, as we saw before, plays a crucial role in bacteria developing resistance to antibiotic, often rendering treatments ineffective. Impact of environmental factors on resistance. Environmental pressure such as antibiotic use significantly influence the evolution of resistance mechanisms in bacteria population. Role of healthcare professionals in resistance. Healthcare professionals are vital in understanding and combating the spread of drug-resistant infection through appropriate prescribing practices. Evolutionary pressure and drug-resistant species included natural selection in microbial population. Natural selection drives the evolution of drug-resistant species where only fittest survive under antibiotic pressure. Contributions of clinicians to resistance studies. Clinicians contribute valuable insight into the mechanism of resistance through research and clinical observation in treatment failures. Effects of misuse on drug efficacy. Misuse of antibiotic contributes significantly to the development of resistant, challenging effective treatment options. Educational initiatives for healthcare providers. Education initiatives aimed at healthcare providers can enhance understanding of resistant mechanisms and improve patient outcomes. Role of genetic mutation in drug resistance. If assume specific mechanism behind genetic mutation, specific genetic mutation in pathogens can lead to changes that confer resistance to therapeutic agents. Influence of researchers on understanding mutation. Researchers are essential in elucidating the pathways through which genetic mutation contribute to drug resistance. Link between genetic diversity and resistance. The genetic diversity within pathogen population play a significant role in the emergence of drug-resistant strains. Patient education on resistant issues. Educating patients about the implication of drug resistance can promote responsible usage of antibiotic and adherence of treatment. Clinical impact of antimicrobial resistant treatment failures. Standard antibiotics become ineffective, leading to persistent or recurrent infection. Increase in mortality, immediate TB, carbamazepine resistant Enterobacteriaceae cereae. MRSA infection have significantly higher case fatality rates, prolonged hospitalization, resistant infections often require 4 antibiotics. Often require antibiotics isolation and longer required. Outbreaks and nosocomial spread. Resistance contributes to the rapid propagation of healthcare association infection. Toxic alternatives. Use of second-line drugs, colistin, linezolid, is associated with how toxicity profiles. Outbreaks and nosocomial spread. Resistance contributes to the rapid propagation of healthcare associated infection. Global burden. World Health Organization estimate 10 million deaths per year by 2050 if resistance is not controlled. Diagnostic and surveillance tools, phenotypic method, disk diffusion, MIC determination, Broth microdilution. Genotypic method, PCR whole genome sequences for resistant genes. Rapid diagnostics include multi-TOF, CRIS-based assays, later flow immunoassays for resistant markers. Global surveillance system, World Health Organization, GLASS, Global Antimicrobial Resistant and Youth Surveillance System, EARSnet, SCDC, European Antimicrobial Resistance Surveillance Network, and SADC Air Lab Network, United States. Pharmacovigilance and regulatory consideration include signal detection. Therapeutic failure reports should be flagged as potential resistant related signals. Increased incidence of resistant infections may emerge from spontaneous reports, real-world studies, or literature monitoring. Risk management plans should include non-resistant mechanisms, target pathogen accessibility profiles, stewardship recommendation. Periodic safety update report prepared must summarize post-market resistant trends, include benefit risk evaluation, and detail emerging resistance signals. Regulatory guidelines exclude European Medicine Agency required American consideration in clinical trial design, guideline on antibacterial agents, 2022. Food Drug Administration emphasizes needs in resistant pathogens, qualified infection disease product designation. ICH A2E recommendation AMR signals be part of pharmacovigilance planning. World Health Organization Essential Medicines List, now stratified by EVERA Access Watch, resort to GIT stewardship. Regulatory tools, companion diagnostic to identify resistant genes, CPERT, MTB, RIF for inflammation resistance and tuberculosis, and conditional approvals with real-world effectiveness follow-up in higher resistance setting. Mechanism of drug-resistant bacteria when we assume and factor contributing to antibiotic resistance in percentage include biopharm formation around 60%, efflux pumps 55%, enzymatic degradation approximately 50%, evolutionary pressure approximately 70%, genetic mutation approximately 65%. Implications of drug resistance for public health include impact of antibiotic resistance, understanding mechanism of resistance, identify the primary mechanism through which antibiotic-induced resistance is crucial for developing effective countermeasures. Public health implications of resistance, the rise of antibiotic resistance poses significant challenge to public health, necessitating new strategies for infection management. Influence of healthcare professionals. Healthcare providers play a pivotal role in mitigating antibiotic resistance through education and responsible prescription practice. Strategies to combat drug-resistant infection. First of all, it's innovative treatment approaches. Developing novel therapeutic strategies is essential to counteract the mechanism of drug resistance effectively. Role of patient education. Educated patient on the importance of completing prescribed antibiotic courses can help reduce the development of resistance. Collaboration among healthcare providers. Collaboration among healthcare professionals is vital to share best practice and research finding in combating drug resistance. Importance of public health policies. Effective public health policies are critical in establishing guidelines for antibiotic use and resistance management. Global health initiatives addressing drug resistance, including international collaboration effort. Global initiatives require collaboration among nations to address the increasing threat of drug resistance through shared resources. Engagement of various stakeholders. Engaging various stakeholders, including governments, NGOs, and healthcare providers, is essential to combat antibiotic resistance. Policy framework for resistant management. Establishing comprehensive policy framework can enhance global responses to the challenges posed by drug-resistant pathogens. Promotion research and development, encouraging research into new antibiotics and alternatives is crucial for addressing the mechanism underlying drug resistance. Impact of drug resistant healthcare system strategies to combat drug resistant infection. You see that it includes public health rates around 23 38%, length of hospital stays around 20%, bacteria mutation rates around 19%, appropriate antibiotic use around 22%, and healthcare costs rising around 17%. When we're talking about antifungal classes, for these classes is characterized resistant to drug resistance. Classes as azoles, fluconazole, voriconazole, traconazole. Mechanism of resistance is RG11 mechanism of mutation. Reduce azole binding to lanosterol, 14 alpha demethylase. Efflux pump overexpression, MDR-Y, CDR1, and CDR2. Upregulation of RG11 or compensatory pathway, and biofilm formation. Fungal species including Candida albicans, Candida glabrata, and Aspergillus fumigatus. Endocannabinoids, casbafungin, micafungin, anilfungin, including FKC1, FKC2 mutation, alter glucan synthase complex, and reduce drug binding, adaptive strand responses, included Candida glabrata and Candida krusei, polynase as antifungal classes, amphotericin B, including mechanism of resistance, ergosterol depletion or alteration, and fungal membrane, oxidative stress resistant pathway upregulated. Bacteria, not bacteria, but fungal species, Candida lusitania and Aspergillus terreus, and flucloxetine, mechanism of resistance include loss of function mutation in cytosine permeasa or deaminase FUR1, Cryptococcus neoformans and Candida spp. And rapid monotherapy resistance can take place too. Clinical impact of antifungal resistance including invasive candidiasis caused by Candida auris, Candida glabrata, show high rates of multi-drug resistance, including resistance to all three major antifungal classes. Azole-resistant Aspergillus fumigatus lead to treatment failure in invasive pulmonary aspergillosis, especially in immunocompromised patients. Increased mortality in resistant fungal infection, invasive fungal diseases with limited treatment option can have cause fatality rates more than 50%. Hospital outbreaks due to MDR strains such as Candida auris, which are difficult to eradicate from healthcare environment and resistant to common disinfectants. Toxicity from salvage therapy, higher dose amphotericin B, or combination therapy may increase risk of nephrotoxicity and hepatotoxicity. Diagnosis oxybility testing, CLC1 AUCAS, both microdilution, attest agar diffusion of MIC determination. Genotypic method, PCR-based detection of resistant genes, RD11, FKC1, FGC2 outbreak tracking, biomarkers, 1,3 beta-D-glucan, halactamanan for diagnosis not resistant. Surveillance program, Fungiscope Global Registry for Invasive Fungal Infection, CDS Mycotic Diseases Branch Monitoring of Candida auris emerging resistant fungi, and World Health Organization Fungal Priority Pathogens List 2022 classified higher risk fungi by resistance rate. Pharmacovigilance and regulatory considerations, signal detection, reporting therapeutic failures, and the guideline concordant antifungal therapy should be assessed for resistance. Spontaneous reports of breakthroughs infection in high-risk population, hematologic malignancies, transplanted, patients with diabetes mellitus may indicate emerging resistance. Risk minimization strategies, labeling updates, emerging resistant patterns, Aspergillus azole resistance in Europe may require revising in the summary product characteristic SMPC. Sur Prebert, drug trends in post-marketing resistance, comparing local and global efficacy in clinical practice. Risk management plan should include non-resistant genes or mutation, cross-resistant potential, antifungal stewardship measures, regulatory agency involvement, European Medicine Agency and Food Drug Administration, encourage inclusion of resistant isolate in pivotal antifungal trials, recognize unmet need designation, orphan status, QIDP for fungal drugs, global health, World Health Organization support antifungal stewardship as part of IMR initiatives. One Health approach emphasizes control in agricultural azole use implicated environmental aspergillus. Investigated biochemical pathway drug resistance. Biochemical mechanisms antiviral resistance include understanding viral mutation process. Viral mutation are a primary mechanism by which a drug resistance develop, affecting therapeutic efficacy significantly. Role of host factors in resistance. Host cellular factors can influence the effectiveness of antiviral drugs, potentially leading to resistant development through various pathways. Investigated resistance in clinical population. Studying clinical population helps identify the prevalence of drug-resistant strains and the biochemical mechanisms, and researching novel antiviral strategies. Developing novel strategies to overcome biochemical resistance mechanisms in viruses is crucial for effective treatment. Metabolic adaptation in resistant organisms is adaptive metabolism pathway in bacteria. Bacterial resistance often involves metabolic adaptation that enable survival in the presence of antibiotics, and impact of environmental stress on resistance. Environmental stress can trigger metabolic changes that contribute to development of drug resistance in various organisms. Cellular response to drug exposure related to resistance. Cellular signaling pathway and resistance. Understanding cellular signaling pathway activated by drug exposure is essential for elucidated resistance mechanisms. Mechanism of apoptosis evasion. Some resistant cells develop mechanisms to evade apoptosis, which allows them to survive drug treatment. Involvement of efflux pumps in resistance. Efflux pumps are vital components in cellular responses that can lead to decreased drug efficacy and increased resistance. Research collaboration drug resistance studies. Collaborative research efforts are crucial for advancing our understanding of cellular responses related to drug resistance. Mechanism of drug in organism we talked about. Again, we see how it's very important. Drug resistant cancer therapy and treatment. Understanding cellular adaptation in tumors. Tumors can adapt to chemotherapy through cellular changes that enhance survival, leading to treatment failure. Patient variability in drug response and individual pattern genetics play a crucial role in how tumors develop resistance to chemotherapy drugs. Biological factors influencing resistance. Factors such as drug metabolism, protein expression, and signaling pathway contribute to chemotherapy resistance. Mechanism of acquire resistance in target therapies. Targeted therapies can lead to mutation in cancer cells that result in acquired resistance, complicated treatment regimens. Impact of genetic mutation on treatment efficacy. Mutation in specific genes can render targeted therapies ineffective, necessitating alternative treatment approaches. Role of a tumor microenvironment in drug resistance. Influence of microenvironment on drug efficacy. The tumor microenvironment can significantly influence the effectiveness of drug by altering tumor cell behavior. Therapeutic targeting of the microenvironment. Targeting the tumor microenvironment presents a promising approach to mitigate drug resistance in cancer treatment. Intercellular communication in resistance. Communication between tumor cells and surrounding stroma plays a vital role in the development of drug resistance. Clinical implication of microenvironment studies. Understanding the tumor microenvironment's impact on resistance offer important insight for optimized treatment strategies. And you see still that mechanism drug resistant cancer treatment is chemotherapy resistant, targeted therapy resistant, microenvironment influence, genetic mutation, and drug efflux mechanism. Challenges in developing drug against resistant strains. First of all, its mechanism underlying drug resistance. Understanding the biochemical pathway that lead to drug resistance can inform novel therapeutic strategies. Challenges faced by researchers. Scientists must navigate complex biological systems to discover effective treatments for resistant strains. Innovative approaches in drug design explore new drug compounds that can bypass existing resistant mechanisms to crucial for effective treatment. Collaboration among experts. Interdisciplinary collaboration can enhance the understanding of resistant mechanisms and lead to successful drug development. Barriers in creating effective anti-resistant drugs. Understanding the biology of resistance. A comprehensive understanding of the biological factors influencing drug resistance is essential for overcoming development barrier. Regulatory challenge in drug approval. Navigating the regulatory landscape is a significant barrier that impacts the timely development of anti-resistance drugs. Stakeholders in drug development. Engage at various stakeholders, including healthcare professionals, is vital for addressing drug resistance effectively. Impact of resistance on public health. The rising prevalence of drug-resistant infection poses a significant challenge to global health initiatives and requires urgent action. Assessing efficacy against resistant strains is the clinical trials focusing on drug-resistant pathogens. Clinical trials must rigorously evaluate the efficacy of new drugs against known drug-resistant pathogens. Engagement with medical researchers. Collaborative efforts among medical researchers can enhance the development of effective treatment for resistant infection. Ethical consideration trials. Ethical challenges arises in conducting trials on population affected by drug-resistant pathogens necessitated careful planning. Drug-resistant mechanism in pathogens include in this way enzymatic dysregulation, target modification, efflux pump activation, reduced permeability, and biofilm formulation. Future direction combined drug resistance. First of all, it's innovative approach to restore drug efficacy. Understand the genetic basis of resistance, exploring genetic mutations that can lead to the development of drug resistance in various pathogens. Collaboration among researchers is crucial. Engaging scientists and clinicians from diverse fields to share insights on combating drug resistance. Investigated alternative treatment strategies. Researching new therapies that may bypass existing drug resistant mechanisms effectively. Research priorities for drug-resistant issues. Identifying care-resistant mechanisms, focusing on the primary mechanisms that bacteria and viruses employ to evade drug action. Global collaboration among health experts, bringing together healthcare professionals to address the challenges posed by drug resistance worldwide. Evaluating the impact of drug use practices, assessing how prescription habits contribute to the rise of drug-resistant strains in various populations, developing rapid diagnostic tools, investment in technologies that can quickly identify resistant strains of pathogens in clinical settings, collaborative effort in global drug-resistant release, unifying research across disciplines, encouraging partnership among academia, industry, and government to tackle drug-resistant challenges, engaging communities in resistant research, involving local population in research initiatives to better understand drug-resistant impacts, promotion data sharing among scientists, establishing platforms for sharing research's findings related to drug resistance across the globe. Clinical impact of patient safety and treatment outcome. Treatment failure increased mortality and infection. Prolonged hospital stays and healthcare costs due to resistant infection and chemotherapy failure. Increased risk of toxicity. when alternative drugs, colistin, linezolid, are required with narrow therapeutic windows. Cross-resistance and multi-drug resistance lead to therapeutic deaths in oncology, psychiatry, infection disease. Psychiatric and neurological relapse due to resistance to antidepressants, antiepileptic mediated by efflux transporters, and receptor adaptation. Regulatory insights and pharmacovigilance strategies. European Medicine Agency and Food Drug Administration demand resistant surveillance data and product development and life cycle management. Resistance may trigger SUR periodic safety data report, prepare update risk management plan revision. The ICH-E2E pharmacovigilance plan and EU GVP Module 3 recommend inclusion of resistant trend in safety specification. I think that it's a model GVP 5 risk management plan, and 7 more closely is periodic safety update report. Pharmacogenomic guidelines, Food Drug Administration table, pharmacogenomic biomarkers now incorporate resistant markers to target drugs. Signal detection, risk minimization in personal drug profiles, integration of real-world data, spontaneous report for emerging resistant trends, decarol resistant in Acinetobacter. Use of machine learning model and pharmacovigilance system to detect resistant cluster or therapy failure patterns. Risk minimization, labeling updates to reflect resistant pattern necessary diagnostic, education material and control distribution, for example, clozapine or vancomycin, diagnostic stewardship company on diagnostic for drug accessibility before prescription, FEGP mutation testing, incentivize for antimicrobial stewardship program embedded into regulatory framework. Dear colleagues, I hope these webinars was useful for you and you can get new knowledge and you can understand how important to taking into account possibility of drug resistance and its role in drug inefficacy. And as I said before, it's now huge challenge and huge economic burden, economic social burden because of real importance of this problem. Dear colleagues, I will be thankful for your question if you will send by my email alina.gmail.com. I will be thankful and very appreciated if you will subscribe to my YouTube channel because I'm trying very hard for everybody, for you, because new knowledge can be very useful, is safe, patient. Today we can be medical doctor, healthcare professional, pharmacovigilance specialist. Tomorrow we can become patient, and very important that this knowledge can spread wise over world, and you can help it, subscribe it, YouTube channel, share this link. Maybe somebody will get this knowledge too. And I will be thankful if you will subscribe, you will visit my website, and I will be thankful for your ideas, what can be interesting for you, your topics can be interesting. I working on books, writing books, and as I see that as I long work, I see how many real problems. First of all, I planned to write clinical aspects of pharmacovigilance. Now I see that every topic can be a topic of a chapter or even book, and who follow me, you can imagine that I already conducted webinar withdrawal syndrome, reborn syndrome. Today it was drug resistance and another very important problem. When we see, when we will go more deeply, we see how important aspirin resistance, clopidogrel resistance. It will be more detailed. Analyze it and show and to write in it with detail. Why? Because too many researchers now we see new one, and it's very important because, for example, when analyzed drug safety in psychiatry and neurology, for example, tricyclic antidepressant, from one side its efficacy of drug, from another side its prolongation of QT, its interval, its problems and sudden death, its problems with withdrawal syndrome, its problems. Why? Because this treatment taken long time and very important to understand consequences of pharmacotherapy. Plus, taking into account, please, its drug-drug interaction, drug-device interaction, and it can be cause of adverse reaction too. I will be very thankful again if you will subscribe on my YouTube channel, plus write comments. It was understandable for you, or you need more detail in which topic, and for me it will help to prepare more useful webinars, also lectures exactly for you. I wish everybody health, happiness, and I want that these drugs will save not one but many patients. Thank you very much. Bye bye.